My name is Jun Li, and I was here almost 20 years ago, 2004 and 2009, and I worked with Mike. I served as my co-chair, and actually during my 5 years here, I worked the entire 5 years with Mike, to the point, I think, looking at Gonzalo was surprised, oh, I'm the co-chair. And so Mike recruited me, and he was, like Sebastian mentioned, so many years as the mission committee chair, and it made a huge difference. I had better offers back then when money mattered so much to me. I got a fellowship over, like $36,000 a year, and I was like, no, I'm going to Michigan. So right after graduation, I joined the University of North Carolina, Chapel Hill, where I'm a professor of genetics and biostatistics, and we're just talking to Beth and many others. We said, like you see, one characteristic of Mike's former trainees, we stay in one place, and I'm in Chapel Hill for almost 17 years, and Beth has been Duke, I don't know, for a long time, and reflecting that, I feel like it's really Mike send us to the best places. He makes sure we're happy and give us all those advice, make sure we're successful. So I'm really grateful. So today I'm going to talk about some of the recent work in my lab along the line of multi-omics understanding of complex human diseases and traits. So as an outline, I will first give a very brief introduction and touch upon two of our recent application work. One is the application of a very classic statistical method called canonical correlation analysis to the top med data. And the other is to predict multiple incident diseases in UK Biobank. So both pieces were recently published. So I'm only going to give two highlights on the interesting findings we have. But if you're interested, I highly encourage you to read the paper, and happy to discuss more offline. And then I will also introduce a method that's still under development. We call it Omega, and I highlighted where the name comes from, and hope I can tell you where each of this letter under the acronym comes from. So as a very brief introduction, we know the power of multi-omics really hold the promise, to teach us about the underlying biology and gaining a deeper understanding of the mechanism behind, and also, ultimately to various different aspects of precision medicine. And we see, explosion of multi-omics, both method development and application work, partly because, many of the projects, including, cohort studies and biobanks, are starting to generate those multi-omics data in really sizable individuals. I feel like even 10 years ago, when I started to read all those papers, and when they claim all of them claim multi-omics, but then if you look at, a number of individuals who have more than one modality, it's pretty much zero. So it's not really multi-omics, but it's really, we, are really blessed with access to different data types. So my lab has been working on this since a few years ago, particularly when, like I said, TopMed, the NHLBI-funded Transomics Precision Medicine program started to generate multi-omics data in real hundreds of individuals, and really hundreds of individuals at multiple points, and the same group of individuals have all those multi-omics data. It made a huge difference. And as I mentioned earlier, we use a very classic statistical method called canonical correlation analysis. And this work published almost 3 years ago was led by my graduate student back then, Min Zhi, and co-supervised by my colleague, Mike Love and Laura Raffield. And using data from two top med cohorts, the Jackson Heart study, JHS, and the MESA study. So both hundreds of individuals with multi-omics data. And so for people who have not heard of CCA, if this is the first time you heard of CCA, it's actually not the first time you use a special form of CCA, because all of us have used a principal component analysis. So CCA, PCA is really a special form of CCA. So what CCA does is actually very simple. So here I have a cartoon illustration of 3 different omics data. So see, this is RNA-seq, proteomics, metabolomics. What CCA does is basically just construct those CVs or canonical variables, which are just linear combinations of the features within each of the omic data, with the goal, to optimize the sum of correlations among the canonical variables. So it's extracting, features that are underlining, multiple of those omics data with the hope to, reveal true biology. So as I mentioned earlier, we have all used the special form PC of it. And as a matter of fact, just to give a fun fact of CCA, it was proposed almost a century ago by someone called Harold Hartley, who spent actually most of his career in Chapel Hill. And in Chapel Hill, we even have a street that's named after him. So you would think such a classical method, and it's just a linear combinations of the features. And the data is not like huge. It's not like a whole genome sequence data. We are having almost millions of individuals. It's just 700 individuals. But this is our first time applying to TopMed. So it's, like I said, for Jackson Heart and Mesa, both, several hundred individuals. On the left are the CVs, top 50 CVs from proteomics data. So this is from the Olink panel. So after QC, we have 1,300, so slightly over 1,000 proteins. And on the right, we have methylation data. So this is like the Illumina EPIC array for people who are familiar. After QC, we have several hundred thousand CPG sites, but actually CCA can, the best implementation back then could not handle all the 700,000 features. So we did some feature selection, just simple ones, most variable CPG sites, and took the top 10,000 CPG sites. And this is what we observe. So we're looking at the correlation among the canonical variables. And you see those huge blocks of red. What it means is those CVs are almost perfectly correlated. So imagine you run PC on your data, and then the software just spill out PCs that are almost perfectly correlated. I was seriously shocked. I mean, even classical method will apply to modern data. So that's actually, one lesson, I think we keep learning, and I really want to warn people who are just starting their career, always be skeptical. Look at your data, do some sanity checks, just to make sure, whatever conclusions you're getting are based on solid, method and results. So seeing that my student Min Zhijiang, implemented this called Grant-Schmidt algorithm, what it does is actually conceptually very simple. It's just, residualize the raw data after each CV, is inferred. So it's you get the first, a pair of triplets of CVs, you residualize using the CVs so that, to reduce. So you see, after the bottom panel shows, after implementation of the algorithm, the non-orthogonality or dependency got much, much reduced. So the only other message I'm going to highlight from this PLOS genetics paper is, a common practice, we need to rethink in our analysis of multi-omics data. So here, what I'm showing on the y-axis are the percentage of variation explained for each of those, phenotype of interest. So it just started as proof of concept, age, sex, BMI. But what I really want you to, focus on are the blood cell traits, so white blood cell count, red blood cell count, and platelet count. So there's two bars. The blue one are for the proteomics, and the purple are the methylation data. So this is looking at how much percentage of variation in each of the phenotype explained by the top 50 CVs. So where you're staring at, this is in Jackson Heart alone, this is in Mesa alone, this is learning the weights from Jackson Heart and applying to Mesa. But you can see very similar patterns. So where you're staring at the numbers, so let me start asking you some questions. So I guess many of you have conducted epigenome-wide association studies, associations of methylation level with various traits of interest. So do you adjust for cell type proportions? Let's say, most likely from blood. Yes, I see a lot of nodes. We do that. That's standard practice. We started almost 2 decades ago. And now let me ask you another question. Will you do associations with proteomics data? Do you do that? No, right? But now if you're comparing the blue and purple, see the blue are consistently higher that means they are actually more associated with the blood cell proportions than methylation data. If it's standard practice to adjust for cell type proportions for methylation association study, why don't we do that for proteins? It's only because we just got the data, right? So we didn't know what, you still don't know the best practices. So yeah, think about that. At least, like in the paper, we seriously advocate, like if you can, by all means. So, I talked about one very classical method proposed 90 years ago. There are many, many fancier methods for multi-omics integration. You see probably more methods than their real data applications nowadays. So, I just highlighted some. This MogoNet published, almost 5 years ago, graphical convolutional network with the view correlation, all those fancy, deep learning methods. And this one, many of you probably have used, at least this version, a particular implementation of the mixed omics package called Diablo, but they have both the sparse canonical correlation based and also sparse partial least square dirichlet analysis, discriminant analysis based. So they have several different implementations. So we actually, like I said, the second piece is also application work. So we basically use the MogoNet mixed omics, particular mixed omics, to try to predict the incidence of 17 different diseases in UK Biobank. So, we are quite familiar with, all the ICD-9-10 codes, but we took the pains to derive the time to event, so to develop those, complex diseases and try to do multi-omics integration. So 4 years ago in Nature Medicine, people have, back then, UK Biobank only had the Nightingale metabolites data. So they tried to do similar things using just the metabolite data that was available back then. So when the proteomics data in UK Biobank became available, we thought, okay, we should do this and see, how much each of the omic contribute and whether, pulling them together help with the prediction of different diseases and whether the conclusion changes for different types of diseases. So that's what we did. And this work was just published actually over the weekend. And my student Jia Wendu did most of the work and co-led by my former student, who is now faculty at Penn, and my colleague, Chen. And so as a brief overview of the study, we started over 20,000 UK Biobank participants. And as I mentioned earlier, we have the metabolites data from the Nightingale panel. So after QC, there's 159 metabolites. And then UK Biobank has proteomics data from the Olink array. After QC, we have almost 3,000 proteins. So for each of the 17 endpoint, again, it's a survival outcome. So we applied the Cox proportional Hazard model to obtain the martingale residuals. And then, as I mentioned, as a primary analysis, we used the package or the method called mixed omics to try to use either just the metabolites alone or the protein alone, all the two omics combined to predict the martingale residuals. And we assess performance using the C index, the higher the better. And then we also did some work to try to see, which of the protein and metabolites are driven, driving the prediction. So that's an overview. Again, I would just highlight, two pieces of findings from this paper. First of all, let's look at this. So again, this is showing the C index. The higher or the redder is better. And each column is a disease. So you can see there's various different diseases. There are cardiometabolic, there's different types of cancer and dementia. And we have 3 blocks on the very, and the 3 blocks differ in the number of the types of baseline covariates adjusted for. So the very top block only adjusted for age and sex, those 2 covariates, and the middle panel adjusted for a more comprehensive panel of 8 risk factors for cardiovascular diseases, and the bottom panel adjusted for 38 very comprehensive panel of risk factors. And then for each block on the top, we have build the model using metabolites only, made one protein only, and the bottom one is using both omics. And so the 2-star means the best performer among the 3, and 1-star means it's the second best performer, but there's no statistical difference from the best performer. So you can see pretty much if we had to pick one, it would be the 2 omics combined. But like you can see across different blocks, the protein omics alone. We should take this with the actual panel. So it's like the Nightingale specific panel and the Olink specific panel, not generalized to proteomics or metabolites in general. But at least in the UK Biobank, the proteomics, the Olink array with almost 3,000 proteins did just as well. And this is particularly true. We are just for a more comprehensive set of clinical predictors. So this is one take-home message. I think when you consider how many omics data shall I generate, probably if you have a limited budget, that's something you should think about whether you really need to generate all the different omics types. And then the only other thing I want to show is regarding the feature importance part. So I just highlighted a few of the cancer findings. So those are top proteins, just looking at the proteins and look at the normalized importance score. So I just highlight 2 findings here. So for prostate cancer, the one with the highest absolute normalized importance score is this K3. For people who don't know this, there's another name. Prostate-specific antigen, PSA. So this is really a positive control. It makes so much sense. And then there's a novel one that we highlighted in the paper, which is this PRG3 protein. It has the highest absolute normalized importance for skin cancer. And this is like a protein related to innate immune response. We actually took it further and performed some genetic causal inference using a technique called Mendelian randomization. So what it does is use the genetic variants or SNPs for the potential mediator, which is this protein here, or in this case, they would be PQTLs, and then try to build a mediation pathway from the SNPs to the protein and then to the skin cancer. And we first used the UK Biobank PQTL results because our finding were from UK Biobank, and we use different methods because there's various assumptions. So I'm going to skip that, but I'm going to highlight is highly significant. We did find highly significant genetic correlation from the protein to skin cancer. And then we say, okay, this is just use UK Biobank data, exactly match what we used for the multi-omics integration. So we try to replicate, use decode PQTLs. They actually use a different panel from a different company. You use a SomaScan proteomics measurement, but we're able to at least nominally significantly replicate the finding, and you notice the direction of association, the odds ratio are in the same direction as well. So this is reassuring. I think this will have some confidence, at least from a statistical point of view, this finding might be real. Of course, we need follow-up studies. So this actually lead to, I talked to you about canonical correlation, about, application using mixed omics, but all of those actually use the data in the formula in the form that we're all familiar with. We call them tabular data. So what are tabular data? This is something we're most familiar with. So it's just like a number of sample by number of feature table, right? So it's a table format. All of those methods just use the data that way. But with the explosion of AI, multi-modal language models, we have realized the power of different modalities. Here, by modalities, I don't mean like a different omics data, but rather different formats, different forms, like a text, audio, image, video, all those type of things. So I just started thinking, why don't we do that? For the data we have, we have those plain, like a boring tabular data. I mean, yes, they can contain a lot of information, but how about considering different formats? Just code them differently so that those advanced methods we have in hand nowadays can better extract the information or extract complementary information. For example, there's a method published about 3 years ago called the genome map. The method actually was initially developed for single-cell RNA-seq data. So you have each row as a cell and each column as a gene. But what this method does is it first constructs a gene-by-gene interaction matrix using the profiles from all the single cells. So basically, they rank the genes in the same order across all the cells. And then what they did is based on the gene ranking according to this gene-by-gene interaction matrix, if you notice the index here, so total we have n genes, and here it's k. So this k is actually square root or close to square root of n. So basically, you convert the profile for each cell or each row here into a 2D information. And if you represent that in heat map format, you get an image. So, basically we take this approach and you can apply the same rationale, to omic data. So instead of having each cell as a row, you just have each sample as a row, right? So that's basically the motivation of our method called Omega, and it's omics multimodality. So the multimodality part, the table format, and I have already introduced you the image modality, and then I will also introduce you the text modality. So it's the multi-omics data converted in different modalities, and then we'll convert using different models to transform them into embeddings. So the graphical parts represent the image modality, and the articulated part basically refer to the text modality. So what's the text? So just giving some examples. So the text part we're still struggling trying to extract the best information because there are so many ways you can convert the information into text, and you can't put everything there. So even for metabolites, for UK Biobank, the Nightingale even with 159, it's not possible because for people who use language models, the token length would be too long for the models to handle. But what we did, for example, one of the things we tried to just take the top 3 features with the largest absolute z-score. So basically normalize across all the individuals. So it's basically for each individual, you find the 3 features that make the most outline across all the individuals. And then we try to keep the original name instead of the acronyms or short names, so that language models can recognize that, and also the original unit again, so that it's more plain English and most friendly to the language models. And then after converting them into different modalities, we use a different model architecture to, because they're the best model for tabular format, for image format would be different. So we choose different models, and then we extract embeddings. So after getting the embeddings, we basically put them into a gating network so that each of the embeddings get their own sets of weights. So this is a very typical mixture of experts, or basically building consensus from multiple different types of models, and then where this is the step where we consider all the different covariates together with the embeddings in their corresponding weights to predict the phenotype of interest. So with that, we actually applied it to the same data that I showed you earlier in our recently published Nature Communications paper. Again, UK Biobank, about 20,000 individuals, about 3,000 proteins, 159 metabolites, the same 17 disease endpoints. And this time I actually used the cross-validation R-square. The C-index I showed also is cross-validation, 5-fold cross-validation based. So we first compared our omega with alternative methods. So omega in red and the other methods in blue and gray. So you can see across all the different diseases by leveraging the different modalities. Although it's the same information, but we just convert in different modalities. It does seem to help. But more interestingly, we performed those ablation studies basically to see like what each of the different modality is doing. So you can ignore the green for now. Let's start with the purple one, which are the tabular format only. And then if you look at just the orange and the blue first, because they are just adding one modality at a time, adding image for the orange part and adding text for the blue part. So you can see, like across different diseases, both of them are, working better than just the tabular data alone. But like which one is better actually differs across different diseases. And what's more interesting, like if you combine the two, it's better than adding each alone. Meaning, again, they are capturing complementary information. It's not like the best one captures, the information in the other two. So again, it's so we also applied to some quantitative traits, blood cell traits, also in UK Biobank, 29 different blood cell traits, and observe actually quite similar patterns. It's not only for the incident diseases, so the pattern pretty much hold at least for the 29 different blood cell traits as well. So as a summary of this work, so again, this is just ongoing work, using mixture of experts and multiple different modalities to code the same data. And we applied to the UK Biobank both the incident diseases and the continuous blood cell traits and observed promising results. And there are a lot of ongoing work. So remember from the Nature Communications paper, I showed you like a PSA. We found the PSA for prostate cancer, positive control, right? And then we have novel findings of, for the driving features. I think that's also a very important part of, multi-omics integration to identify which of the features are driving, predicting, or sometimes it could be like early on serve as biomarkers. So in a mixture of expert framework, it's actually not that straightforward. So that's, again, some ongoing work. And with that, I'd like to thank people who have been leading this work. So my student Wenhao Zheng and also Jia Wen, who led the previous application work as well, and my other student Mu Qing, and this is co-supervised with my former student Chen Sun and my colleague Hua Xiu Yao, and also thank many people in my lab. And this is a picture we took last year at ASCGG. We try to have those gatherings at ASCG. But guess where this heritage started? I mean, this is my first year attending ASCG. This is my second year of my PhD, and. Even in my second year, I didn't even have a poster. The only reason I didn't go in my first year, 2004, was it's in Toronto. And I had some visa issue. And my eyeballs almost dropped when Mike said, oh, you should go. I was like, I don't have anything. But Mike has always been so generous, sending people to conferences and all the network. It's really, we benefit lifelong. And this is over 20 years ago. It took me quite some time just to dig out all those pictures. And and actually most of the time it's a big group of us, having fun, both scientifically and just as a group hanging out together. It's just so amazing. And then I was reflecting, looking at the projects, I feel, okay, I do things so much different. I do, relatively, I still work with Nancy a lot, PRS and all those more traditional statistical genetics, but I do much less of those. I was like, oh, what have been done? But then I was, going back and reflecting on my very, very first project I, did with Mike. That's actually before school even started in 2004. So I was, I did my master's at Bowling Green State University, which is 17 miles from Ann Arbor. So I came here, I think, 2 or 3 days each week. And this is the very first project I worked with Mike. And I presented the results, when school started. And this is looking at the association between coffee's consumption and the risk of type 2 diabetes. And back then, even using cross-sectional data, we didn't even have prospective data. But it was so much fun, just exploring different things. So I was like, okay, I should not feel that guilty not doing a lot of, like a classic, statistical genetics. Because see, my first project with Mike almost had nothing to do with genetics. But I can't, talk about my graduate studies without mentioning this paper. So can you imagine your first papers being a science paper as a grad student? I mean, it's just so amazing. So we started to have those GWAS data. So I think Richard just mentioned 1996, the Rich and Americanga's science paper talking about association studies. But it's not until a decade later we started to really have those GWAS data, and we have over 2,300 individuals, about 1,200, cases and controls from the fusion study back then. It's an Illumina 300K panel. And Mike had to buy a new computer just to run Genome Studio on that. It ended up as a paper. It was a lot of work doing all the careful QC, and I remember learning so much from Anne and Heather. All the QC and various different sensitivity analysis. And the other thing is towards the end of a manuscript putting together. I think that's when Mike started forming the spring break in Hawaii tradition, right? And so I remember in and Heather's office, I was like, isn't Mike in Hawaii? Why is he having so many emails? On the manuscript, because it's around the time we're kind of starting the manuscript, and it was like, oh, if I were in Hawaii, I won't work. That was so wrong. I've been in Hawaii 3 times, but I worked every time I was there, out of my own view. It's really, and so I really want to highlight the last part, a quote from Benjamin Franklin. Involve me and learn. There's so many things Mike has involved me. I still have papers which I'm only very peripherally involved. Even last year on type 2 diabetes, and I can't even count definitely over a dozen diagram gent papers. Just Mike involved me from the very study. It's a really learning and a collaborative experience. All the network, not only science. It's so much fun. And I like to end with this quote, and it's really something. Mentor is someone who sees more talent and ability within you than you see in yourself and helps bring it out of you. Thank you so much, Mike.